How's it going, everybody? This is Mr. Thompson, and I'm going to take you through the DNA extraction lab, the cheek cell extraction lab, in case you weren't able to join us in class. Okay, so I'm going to go through all the procedures that I have listed in the lab handout. So you want to make sure that you've opened up the remote edition, okay, and have that open somewhere so you can refer to it as we're going through. Okay, so I've got my materials here. Now I'm using my test tubes, but you'll use whatever vial you have from a science kit or a pool cleaning kit or something like that, okay? And I have my cup and my sports drink that I'm gonna use. I've got my detergent solution that I'm gonna use. Now I've already pre-made these, okay, but you wanna go back through and you wanna look at the procedures on how to do the detergent solution and the alcohol solution. This is my rubbing alcohol, okay, and it's at room temperature. I didn't chill it like I did with the other alcohol that I'm using in class. Also, I've got my medicine droppers here. And I've got different medicine droppers that I want to use so I don't have, contamination and stuff like that. So, first thing what I'm going to do after I get my test tube, which I have right here. Also, you want to have some sort of something to hold the test tube up, like a cup will work just fine. I actually have a test tube rack that I'm going to be using over here off camera. So I'm going to take my sports drink. I did red fruit punch, even though it's not my favorite color. But one of the things that's pretty cool about this is if you do it with the different color sports drinks, your sample turns that color too. And I think that's pretty neat. So I'm hoping that I get a really good pink DNA sample. So that's why I'm using this one. Blue works, yellow works. Heck, you can do it with salt water and you still get it done. This is also tastier. So I am going to, I've got my cup and I'm gonna pour, this says 8 milliliters of sports drink. Now 8 milliliters is like an ounce or so. If you have the little medicine cups that come with children's medicine, like children's Benadryl and stuff like that, you can do, put it halfway in between the 5 and the 10, unless you have one of the fancy ones that have all the individual markings. I don't have that, I didn't bring it with me, so I've got my cup. And so I'm gonna fill it like that. You don't need that much, okay? Because you can overfill, you don't wanna overfill your test tube, right? So I've got this. Now I'm going to swirl it. Now when I swish it around in my mouth, right? Scrape the inside of your cheeks with your teeth to get some of those extra cheek cells. The more cheek cells you have in here, the more DNA that you can get, all right? So, bottoms up. Okay, so I've mixed my stuff and I spit it back into the cup. I guess I should have warned you. You should know this anyway because you read it. Okay, so I've got this. I'm gonna pour all of this into all of this. Now, if you have one of the wax paper cups, you can actually pinch off this part right here to make a little pour spout. If you have the plastic cups, you can kind of do that, but then it breaks like that. So just be real careful when you're pouring it in. Now as you can see, I've got some left over. I ran out of space. So I poured in as much as I could, okay? So now we have our mixture right here. Now we're going to take our detergent solution, right? And so remember, this is just soap and water, okay? And so what we want to do is the soap is going to break open those cells, exposing that DNA, okay? Because right now the DNA is snug in those cells. and we can't get to it. So we're going to open that up for us. So I'm going to take my solution. Now it says about 2 milliliters. If you have a dropper like this or a regular dropper, you know, a couple of dropperfuls will do it. I'm gonna kind of have to be careful because I overfilled mine just a little bit. You can kind of see it. You want to do it down the side so we don't disturb our solution too much. I'm just gonna put the whole thing in there as much as I can fit in there. All right, so I've got my stuff in there. It's mixing. Now, what we want to do is we want to cap the tube. So if you're fortunate enough to get one of the plastic vials for like science kits or pool supply companies for pool testing chemicals, just screw the cap on there. I'm not that fortunate. I've got an empty top, but it's my spit, so it's not gonna hurt my feelings that much. I'm gonna cap it with my thumb. I'm gonna invert it 5 to 8 times. And so when you're inverting it, you wanna go completely upside down like this and let it mix. Okay, so you're gonna do this 5 to 8 times. 3, 4, 5. I'm gonna go ahead and go all the way to 8, 6, 7, 8. All right. Hopefully that did that. Now, what I'm gonna do is I'm going to add my, oh no, before I do that, now I'm gonna let this sit for 2 minutes. So I'm gonna set myself a timer right here for 2 minutes. See how it's going down? I'll see you in about 2 minutes.
2 very boring minutes later.
Okay, so 2 minutes has gone by and we've let our detergent solution a chance to mix. That's gonna break open those cell membranes and allowing that DNA to come out, right? And so now what we're gonna do is add our alcohol solution, right? And so now, if you read the beginning stuff, that intro stuff at the beginning, you know that DNA does not dissolve in alcohol. And so when we add the alcohol to our solution, it's gonna cause the DNA to precipitate out. And that's just a fancy word for saying it's no longer gonna be dissolved in the liquid, it's gonna be solid and visible so we can see it. And that's kind of what we're going for. So I'm gonna take my alcohol right here. I've got my test tube right here. I'm gonna add a little bit of alcohol, enough to cover the top and make a layer. You wanna shoot for 1/2 to 3/4 of an inch. I don't have that much space, so I'm gonna do the best I can. Okay, I've got a little bit of alcohol in there, and now set yourself a timer. We're gonna come back to this in 15 minutes.
15 minutes later.
All right, so 15 minutes is up. Now I ended up going in, almost forgot. All right, so I ended up running, I ran it twice because that first one was real full, so I wasn't sure if I was gonna get anything from it. So here's that first one right here. You can't really see anything from it, right? Let me see if I can focus on that one section right there. Can't really see anything with that one, which is okay. And I will say that not everybody's gonna get everything from this one, so don't be surprised if you don't see anything. However, this one has some liquid, wrong way, has some liquid in it. Not liquid, it's got some DNA in it. Let me see if we can focus in real quick on just that. It's kind of hard to tell. It looks like stringy stuff. Tell you what, I'm gonna take a picture of it. I'm gonna pop it up right here. Okay, so here's that stuff again. Here's a little bit of a better shot of that stuff. And you can kind of see that sort of stringy stuff that's in there. That is the DNA. Okay, so now we've gotten some DNA in there. What we're gonna do is I'm gonna get another clean medicine dropper. Okay, so here's my clean medicine dropper. I got it from my box of goodies right there. And I'm gonna stick it into this upper liquid part right there. And I'm gonna suck up the stringy strands of DNA. Okay. Pull that out. Set that down. And now I have these little micro centrifuge tubes. So these teeny tiny little vials right here. And I'm gonna squirt my DNA in there. Let me pull up a little bit more stuff just to. And actually, I think what I'm going to do is I'm going to put in a little bit more of that rubbing alcohol stuff in there just to make sure everything separates nice and neat. Close the cap, nice seal, and there it is. That's my DNA right there. Sort of what makes, yeah, that's a little bit better. Sort of what makes me, all sort of bunched up into that little vial right there. Take a picture of it. And that's it. You have extracted your DNA from your cheek cells. Now this also, you may have done it when you were younger. Something like that. You did it with strawberries, okay? Same basic principle, a little less mess. So yeah, use this, drop that picture on that lab there. If you have any questions as you're going through, taking a look at it, let me know. The questions that you're answering are the same questions as the folks in class. It covers all the same material and stuff like that. Go back and look at this. If you run into any problems, or you can send me an email or something like that, and we can troubleshoot what's going on and stuff like that. And I hope you have good luck. I look forward to seeing y'all results, and I look forward to seeing y'all again soon.